All right, we are on chapter 6, section 6. So this one, we're going to start talking about polysaccharides. So we're going to start linking together our sugar units, right? So we know that glucose is basically our source of energy for everybody, plants, animals, everybody. But think about, like, plants are the ones that generate all of our energy from solar energy, right? And they turn it into chemical energy. But what do they do at night, right? They can make their own food during the day, but they can't make their own food at night. And so if they didn't have a way to store that food, they would die at night. So glucose can be stored in plants, and then animals can also store glucose. So we store it a little bit differently. In plants use starch to store their glucose, and we use glycogen. But whether it's starch or it's glycogen, you're connecting individual glucose units. And those individual glucose units are alpha glucose. So remember what alpha is telling you, that is a trans orientation, and that's through glycosidic bonds. So you can connect many beta glucose units, and this produces something different. This produces cellulose. And cellulose is a structural material, right? So do you want to use it for food, or do you want to use it for structure? If you want to use it for different things, they're going to be linked differently. So remember that storage is alpha and structure is beta. So these polysaccharides, remember we said polysaccharides can be like huge, right? They're large, large molecules, and there are many, many, many monosaccharides bonded together. So the polysaccharides don't have enough reducing ends to give a positive Benedict's test. So remember our Benedict's test, we wanted to see if there were reducing ends. So you can tell if there was sugar in like somebody's urine or something like that. But when we talk about polysaccharides, right, they're in such a long chain. There are so many of them that you only have the very ends that are reducing ends. And remember, a reducing end means that you have a free anomeric carbon. So that carbon that's attached to two oxygens. And so because the large portion of the molecule is all connected, because they're really, really, really long chains, even though you do have reducing ends, you're not going to get a positive Benedict's test. All right, so let's talk about these in a little more detail. There are two different storage polysaccharides for plants. Starch is a mixture of these two. So we say that plants store their glucose as starch, and starch is composed of two different polysaccharides. The first polysaccharide is called amylose. Amylose makes up about 20% of the total starch, and this has a straight chain of 250 to 4,000 D-glucose units. Remember, everything's D form because that's what we can handle. And they're all linked through our alpha 1,4 linkage. That's really, really important that you know that, alpha 1,4. So when this happens, we say it's a straight chain, and that's true. It links one after another, after another, after another. But because of the way it's structured, that amylose is going to actually begin to coil. And so it looks almost like a spring when you look at just amylose. Now, it's only 20% of the starch. The rest of the starch is called amylopectin. And it also contains D-glucose, and it's also connected by an alpha 1,4 glycosidic bond, so that's the same. But what's different here is that about every 25 glucose units, there's a branch. So you have glucose unit after glucose unit after glucose unit, and so on this really long chain, you're going to have a branch come off. And so this branch is through a different bond, because you can't branch off of the same bond. And that's an alpha 1,6. So this is the branch. The branch is our alpha 1,6 linkage. And we're going to look at some diagrams. So when we are hungry, we eat plants, right? And the plants contain that starch. And then our digestive system can break down the starch into individual glucose units that our bodies can use. Thank you, plants, right? So here's our structure of amylose. So remember, amylose is our straight chain, right? And it has that alpha 1,4 glycosidic bond. And so you have them, here's kind of the zoomed in, right? We know, whoa, that we're numbering our carbons based on the carbon that's attached to 2 oxygens. So this is carbon 1, here's carbon 1, 2, 3, 4. And then we see that this is a trans configuration. So a trans tells us that it is alpha. And so that's an alpha 1,4 glycosidic bond. And it's just repeated over and over and over and over and over and over. And what happens is that this ends up being a coil. Even though it's just one chain, the 3-dimensional structure turns it into a coil. Okay, so that's amylose. Here's amylopectin, right? In amylopectin, what we have here, we have those chains, and then here, oh, we're going to have a branch point. So where's our branch point? Well, here's our oxygen, so that's carbon number 1. And over here, that's 1, 2, 3, 4, 5, 6. So this is, and it is again, if we check here and we check our oxygen, we're trans. So this is an alpha 1,6 glycosidic bond that makes the branch. Right? Our chain is still a 1 and a 4. It's still our alpha 1,4, but the branch is an alpha 1,6. So make sure you know the difference between the branch and the straight chain. Okay. So our bodies can also store energy. And we talked about this in class where we said, you know, if you go to sleep and you don't eat for a really long time, you're not going to die because body stores sugar. And where does it store it? It stores it in the liver. And we call it glycogen. So glycogen is another example of a polysaccharide. And glycogen is identical in structure to our amylopectin. So all of the linkages are the same. The branches occur in the same place. But what's different is that amylopectin branched every 25 units, whereas glucose is going to branch every 12 units. That's the only difference. So if you have these stores of glycogen in your liver, they're going to maintain that storage. And then when your blood sugar gets low and you don't eat, then your body's going to send a signal through glucagon to say, hey, liver, you need to cut up some of that glycogen and put it out into the bloodstream. And so because you have so much branching in glycogen, it's really, really quick and easy to cut a little bit off and send it into the bloodstream. So here's your kind of summary, right, of the 3 different storage molecules. We have amylose and amylopectin. These 2 are in plants. Glycogen is in animals, right? Amylose is that alpha 1,4 chain, and it's really long. It's 250 to 4,000 units of glucose. Amylopectin is the same alpha 1,4 linkage between them, except we have a branch on the alpha 1,6 every 25 glucose units. Glycogen looks just like amylopectin, except what's different? Here, amylopectin's branching every 25 units, whereas glycogen is branching every 12 units. Okay, all right. So now, we said there were 2 different kinds of ways that you use polysaccharides. One is storage and one is structure. So our structural polysaccharides, one of them is called cellulose. Cellulose is a different glycosidic linkage. So remember over here, our glycosidic linkage was an alpha 1,4. Here we are a beta 1,4. So how important is that cis or trans orientation of that extra oxygen that sits on the anomeric carbon? Very, very important. So just because of that one little bitty old change in our glycosidic bond, it's going to completely alter our overall structure of cellulose. So when amylose was coiling, right, because we have this beta linkage instead of the alpha linkage, you're going to have straight chains, and they're going to be super, super straight. And they actually end up lining up one on top of the other, one on top of the other, on top of the other, on top of the other. And you can think that because they align so close to one another, they can form really, really rigid and strong structures. And we're actually unable to digest cellulose. It's an insoluble fiber, right? But we still need to consume it because it helps to assist movement within the small and large intestines. And so what you can see here, this is our little diagram of cellulose, right? And so you can tell here that you have, here's our carbon-6, and then we have a beta linkage, right? Because they are cis, we have a beta linkage. So it's still a 1,4, right? This is 1, 1, 2. Ooh. This is, if I could do this right, 1, 2, 3, 4, right? So that's still our 1,4, but it's a beta linkage instead of an alpha linkage. And so these end up just like this, super straight. And then you have another row underneath and another row underneath, and it gives lots and lots and lots of stability. So chitin is an example of another polysaccharide that's structural. So chitin is very similar. It has our glucose molecules, but those glucose molecules are a little altered. So we still have our beta 1,4 linkage, but what's different is at the bottom of our ring, right? If you're on 1, 2, usually coming down here, you have an alcohol, an OH group. But instead of having that alcohol OH group, what we have is a nitrogen, and then that nitrogen is bound to, this is called an acetyl group. This was one of your functional groups. So this is our acetyl group. And so we say that this acetyl group is bonded to the glucose through an amine, right? This was another one of your functional groups. An amine is a nitrogen, right? And depending on if it's a primary or secondary, how many things it's attached to, but it's still an amine. So we have 2 extra functional groups in chitin, okay? And I'm gonna show you, you have these pictures, you have this picture here. I just kind of copied and pasted, but you still have that picture. So if you want to go in and add it on there, you can. All right. Okay, so our N-acetyl, we call it an N-acetyl glucosamine. So N for the nitrogen, acetyl, and it's on a glucose, and it's an amine. Okay, so N-acetyl glucosamine is our chitin molecule, right? So like cellulose, chitin's very, very strong, and it's used in insects' exoskeletons because they're super, super hard. And it's also waterproof, which makes it really, really interesting. And you can exploit that property in waterproofing many different things like water. We also use it as a surgical thread, and we use that because the body will break it down. It's something that the body can actually metabolize. And so that's our dissolvable or biodegradable sutures, which is really cool. So if you grind up chitin, then this is a powder that can actually hold in moisture. And so you will find chitin in makeup and lotions and things like that. It'll keep you hydrated, right? It really will. All right, so you have to know your 4 different kinds of, well, it's actually 5. It's actually 5 because we have amylase and amylopectin, right? And you know, these were from plant. And then you had, you also had, and I'll kind of stick it in here, in animals, you have glycogen, which is just like amylopectin, right? Except it doesn't have, it has the rings every 12. You have a branch, let's write that again. You have a branch every 12 units instead of every 25 units. So this branch every 25 units, okay? All right, and then you have that's storage, right? So all of these up here, all of these 3 up here, these are storage for energy use. These two are structure. So structurally, we talk about cellulose and we talk about chitin, okay? So you need to know the difference between those. All right, so identify the polysaccharide described by each of the following. A polysaccharide that is stored in the liver and muscle tissue, that is going to be our glycogen. A component of starch containing only alpha-1,4 glycosidic bonds. Well, if we are a component of starch, there's only two components of starch, amylase and then amylopectin. And one of them contains only alpha-1,4 glycosidic bonds and the other one has branches. So which one does not have branches? That is our amylase, AM. Okay. A polysaccharide found in the exoskeleton of insects. And that one you should remember is chitin. C-H-I-T-A-N, C-H-I-T-I-N. Whew, I-N. Okay, all right, let's go to a few quick problems. So I forgot, we're not doing the problems on 6.6. We're gonna do them in class because there aren't that many. And I wanna cover 6.7 in this video as well because it's relatively short. All right, so when we talk about carbohydrates, right, we usually think about things that we eat, but we use them for other things inside of our cells. We use them a lot of times in cell signaling. So our cells, we have to have a way to know our cells from foreign cells. So think about if you have an infection, right? You want your body to be able to recognize that bacteria or that virus and kill it, while at the same time recognizing your own cells and saying, oh, that's me, it's okay, I'm gonna leave those cells alone. And so this is really important when we talk about things like blood types, because you need to be able to have a mechanism where your body can not react to your own cells. If you have an immune system that attacks your own cells, you have an autoimmune disease. And so that can be very, very dangerous. So we have to think about that when we're using modern science in terms of donating blood to people, right? So red blood cells, just like every other cell in your body, have markers on the outside of the cell surface. And so we call these, there are a couple of different kinds, but the most important are called the ABO blood markers. And the ABO blood markers are little carbohydrates that stick out on the outside of your cell, and they're unique. So they're unique to your blood type. And so, you know, you're told you have type A, or you have type B, or you have type AB, or you have type O. What is that telling you? It's telling you the combination of monosaccharides that you have on the outside of your cell surface. So there are only 4 different monosaccharides that are present on the outside surface of your red blood cells for the ABO blood type. And those are D-galactose. Now look at this. This is kind of cool. L-fructose. We almost never see the L configuration, but we actually do see it in this one. So this is L-fructose. That's kind of cool. Then you have N-acetylglucosamine. So remember the acetyl, right, is telling you that you have an amine group. So the amine is right here, because you have an amide group right there. And then that connects the acetyl group. So here's your acetyl group, right? And then it's glucose. So N-acetylglucosamine. And then you also have N-acetylgalactosamine. Galactosamine. Okay, so our amine is the nitrogen. Our acetyl is right here. And then the original structure was galactose. And so that's why they're just saying you're modifying that by adding an amine and then adding an acetyl group, okay? So what does that have to do with our blood types? Well, here we go. So if you are type O, you have 3 different kinds of monosaccharides inside of the chain that's sitting on the outside of your cell. So you have an N-acetylglucosamine, you have a galactose, and you have a fructose. So what you'll see is that these three are present no matter what type of blood you have. If you are type A, you have N-acetylglucosamine, galactose, and fructose. But you have something else also. If you are type A, you also have an N-acetylglucosamine, right? So this is my extra thing that type A has. If I go to my type B has the same three as type O, N-acetylglucosamine, galactose, and fructose. But like type A, it has something extra added to it. What is the extra thing that's added to it? The extra one is our galactose. So this is unique to type B. We have galactose. Now, what if you're type AB? Well, if you're type AB, then you have a red blood cell. So here's your RBC, here's your red blood cell. And your red blood cell has both the type A sugars and it has the type B sugars. So type AB is just saying that you have both the N-acetylglucosamine, galactose, fructose, galactosamine, right? And then you also have the N-acetylglucosamine, galactose, fructose, and galactose, okay? So it's just telling you what's on the outside surface of your red blood cells. So those are our four types: type O, type A, type B, and type AB. So this is type AB, has both, okay? So when we talk about donating blood, what we have to worry about is, if let's say I am type O, right? And they want to give me a blood transfusion. If you give me blood that has antigens on the outside, and these are called antigens, but they're really carbohydrates. If it has carbohydrate markers on the outside of those red blood cells that I'm going to receive that I don't recognize, then I'm going to say that's foreign, and my body's going to have an immune response, and I'm going to reject that blood, and then you could potentially kill the patient. So it's really, really, really important that you give the patient the right kind of blood. Okay, so let's talk about the different ones, right? So each person's immune system can only recognize its own carbohydrate set. If you're type A, you're gonna look at things that are like A, and you're gonna say, okay, these are okay. So if you're type A and you saw some type O blood, oh, that looks like me, that's fine. So you would be able to tolerate type A blood. But if somebody gave you type B blood, oh my goodness, look at this galactose, your body would say, no way, that's foreign, I have to get rid of that. So if you were type A, you could not receive type B blood or type AB blood. So it's very, very, very important. So because the trisaccharide that's on O is on all of our other blood types, so here's the trisaccharide, that's the one that I've circled in the square, in the rectangle, here, here, and here. So look at that, all the blood types have those three, that trisaccharide sequence. And so all of the blood types, whether you are O, whether you're A, whether you're B or AB, those can all receive type O blood because they're not going to recognize that carbohydrate set as foreign. So type O is our universal donor. So you could say that type O is the universal donor, right? The type AB blood is considered the universal acceptor, right? So if you go back real quick to type AB, let me kind of erase this stuff, right? Type AB has all of this on the outside of its cell, all of this. So type AB can receive blood from anyone because it looks at all that stuff and says, hey, yeah, that looks like me, it's cool, right? If it gets type O blood, well, that's fine. It's got this stuff on there. I'm okay with that because I have that on my cells. All right. What if they get type A blood? So they're going to get markers that look like this. Well, that's already on my red blood cells, so it's fine. What if I get B blood? These are the ones I'm going to see on that transfused blood, and it's okay because that's on my red blood cells, so I'll be fine. Okay. So type AB is called the universal acceptor. It can be, that person who has type AB blood can be transfused with any other blood type, doesn't matter, does not matter. Okay, so this was a nice little table they did to organize for you. So they listed each of the blood groups, your A, B, AB, or O. And they tell you who you can receive from and who you can donate to. So if you're type A, right, you can receive from anyone who is just like you, or somebody who has the same carbohydrate set minus a piece, right? Remember A, we had our 3 carbohydrate chain. If I can draw this real quick. Ooh. But that's your 3 carbohydrate chain. And then you had that one thing that was a little bit different, a little bit extra that was coming off, right? And you have your extra carbohydrate there. So A can receive from A because they match, they look exactly the same. Well, how about O? It can receive from O because O is just this chain right here. And so the body says, oh, well, that's in my blood, so it's okay, I can receive that. B, on the other hand, B has exactly the same thing. I'm just going to copy and paste, except my extra little thing right here, that is going to be a different extra group, right? So it's an extra group, but it's a different kind of carbohydrate. So let's stick it on a different color, right? So now I can see that B can also receive type O blood because, again, it has that same sequence of that trisaccharide. So B can receive O blood. AB. AB has both of what is in A and B. So I'll put it, right? I'll put it right there. That's what AB, this is AB. AB is both of all of that stuff. So it can receive from anyone. And then the last one is type O blood. What is type O blood? Let's see if I can do this. Type O blood is just the 3 carbohydrate chain, right? And so it can only receive blood from itself, from that same blood type, from O. Can't receive from AB, can't receive from B, can't receive from A, because all of those blood types have that 4th carbohydrate on it, and the body would recognize that as foreign and attack it, and so they would be in trouble, right? All right. The other important carbohydrate that we're going to talk about in terms of blood is called heparin. Heparin is an ionic polysaccharide. And it's an ionic polysaccharide because it has many, many disaccharide units. And so I know they're only showing 2, but you have to kind of remember that there are going to be additional monosaccharides, disaccharides, excuse me, right? And then you'll have an O, then another, I'm not very good at this, but so another one, and another one, and another one, and another one, right? And so it's ionic, it has a charge. So you see all of these things here that have a charge, they're ionic, and this one too. And that allows it to prevent blood from coagulating or clotting. And so we call these glucosaminoglycans, and they're charged. And so that's what gives them their unique property to prevent clotting. And it's because it has a sulfate on it. So the SO3 is the sulfate, SO3, and that gives it the majority of its charge. And so a lot of times you'll hear people are given heparin if they're maybe at a very, very high risk of having a heart attack, like maybe they're having a heart attack. You need to give them something so that their blood won't clot. Heparin's also used if you're going to do a blood draw. The vial that the blood is going to go into a lot of times is coated in heparin so that when the blood is pulled from the patient, that it doesn't clot inside of the tube before you can go and get it tested and that kind of stuff. Okay, and then we're going to do one little problem here, and then we'll do the rest of the problems in class. All right, explain whether the following blood types could be donated to a person with type A blood. So you have a little chart here, but you're not going to have this chart on the test, on the exam. So this you have to be able to understand and explain. So we want to know, could the person donate? So in A, we have a person who's type A, and we're going to donate to someone who is type A. So this is going to be the recipient, right? See, E, recipient, right? Okay. So if you are the same blood type, you can always receive from yourself because your carbohydrate looks the same. So this is A-okay. How about B? We're going to donate from type B to type A. So here's my recipient right here. This is my type A person. They can receive from anyone who looks exactly like them or anyone who has one of those extra carbohydrates missing. Like if you have somebody who doesn't have this, they'll only have the 3, then that would be somebody who is type O. So you can only receive that way. So if somebody who is type B, so type B is here, if I give this type B blood to somebody who's type A, the cell's gonna look at this galactose right here and say, whoa, stop the train. That I can't accept that. That is not self, that's not me. And so it must be foreign, and they would reject it. So this would be very bad. You would have a very bad reaction. Okay. How about C? C is type AB going to A. Well, if it was just type A, we'd say, yeah, okay, no problem. But it's the same thing here as in type B. The person who's AB has this stuff sitting on the outside of their cells. So if you gave it to somebody who's type A, again, you would have a rejection. And so it would be very, very, very bad. All right. And the last one, D, we're type O going to type A. So that means that we are this blood type trying to give to this blood type. And you can do that because type O has this saccharide grouping that is also in type A. And so it's okay for type A to receive type O blood. That's why you hear them you'll hear them say, hang a bag of O neg, and it's O negative. Negative is another grouping that we're not going to talk about. And type and cross their blood. So if somebody comes into the emergency room, they don't know what their blood type is, they're just going to they're going to give them some O negative blood. And then in the meantime, because you don't want to use up all your O negative blood, you're going to take a sample from the patient and you're going to determine their blood type. And that's what's called the type and cross, right? So you're going to determine what their blood type is. That way you can go in and say, okay, well, if they're type AB blood, they can get any blood type. And so you don't have to use up all of your O negative storage. All right, that is the end of chapter 6. So see you in class. Have a great day.